Nitric oxide receptors in hormones in detail. Very long answer. ChatGPT. Nitric oxide (NO) is a signaling molecule that plays a crucial role in various physiological processes, including vasodilation, neurotransmission, and immune response. While NO itself is not a hormone, it acts as a signaling molecule that influences hormone release and other cellular functions. NO does not bind to traditional cell surface receptors like other hormones, but it exerts its effects through interactions with target proteins and second messengers. Let's explore the details of how NO influences hormone action. NO signaling pathway. NO synthesis. NO is produced by the enzyme nitric oxide synthase (NOS), which converts L-arginine into NO and citrulline. Diffusion. NO is a small and highly diffusible gas that can cross cell membranes and travel to neighboring cells. Activation of guanylate cyclase. In target cells, NO binds to an activated soluble guanylate cyclase (SGC), an enzyme present in the cytoplasm. cGMP production. Activated SGC converts guanosine triphosphate (GTP) into cyclic guanosine monophosphate (cGMP). cGMP signaling. cGMP serves as a second messenger that mediates the effects of NO. It activates protein kinase G (PKG), which phosphorylates various target proteins and ion channels. Influence of NO on hormones and endocrine functions. Vasodilation. NO is a potent vasodilator that relaxes vascular smooth muscle cells, leading to increased blood flow. This effect can influence hormone delivery to target tissues and affect hormone clearance by altering blood flow through organs. Neurotransmission. NO is involved in neurotransmission by modulating the release of neurotransmitters such as dopamine, serotonin, and acetylcholine. This can affect hormone release and signaling in the nervous system. Insulin secretion. NO has been shown to influence insulin secretion from pancreatic beta cells. It enhances insulin release by stimulating cGMP production and activating package. Hypothalamic pituitary adrenal (HPA) axis. NO can modulate the HPA axis, which controls stress responses and hormone release. It can influence the release of hormones like cortisol through interactions with the adrenal glands and the hypothalamus. Immune response. NO plays a role in immune responses by modulating the release of inflammatory mediators and influencing immune cell function. It can affect the production and release of cytokines and other immune-related hormones. Endothelin regulation. NO can inhibit the release of endothelin-1, a potent vasoconstrictor hormone produced by endothelial cells. This helps maintain vascular tone and blood pressure. Reproductive functions. NO is involved in regulating reproductive functions, including hormone release and blood flow to reproductive organs. Therapeutic implications. The role of NO in regulating various physiological processes has led to the development of drugs that target NO pathways. These drugs are used to treat conditions such as hypertension, erectile dysfunction, and cardiovascular diseases. In summary, while NO is not a hormone itself, its role as a signaling molecule has significant implications for hormone release, signaling, and overall physiological functions. NO influences hormone action by modulating second messenger pathways, vascular tone, and neurotransmission, among other processes. Its intricate interactions with the endocrine system highlight the complexity of cellular signaling and its impact on hormonal regulation. G-protein coupled receptors, steroid receptors and nitric oxide receptors in hormones in detail. Structure and examples. ChatGPT. G-protein coupled receptors, GPCRs. Structure. GPCRs are integral membrane proteins composed of 7 transmembrane helices. They have an extracellular N-terminus, an intracellular C-terminus, and 3 intracellular and 3 extracellular loops. The binding of hormones to GPCRs triggers a conformational change that activates intracellular signaling pathways through G-proteins. Examples. Examples of GPCRs include the beta-adrenergic receptor activated by adrenaline, the rhodopsin receptor involved in vision, and the angiotensin II receptor involved in blood pressure regulation. Steroid receptors. Structure. Steroid receptors are nuclear receptors found in the cytoplasm or nucleus. They have distinct domains, including an N-terminal domain, a DNA binding domain, a hinge region, and a ligand binding domain. Upon hormone binding, steroid receptors undergo conformational changes and translocate to the nucleus to modulate gene transcription. Examples. Examples of steroid receptors include the estrogen receptor (ER), the androgen receptor (AR), and the glucocorticoid receptor (GR). Nitric oxide (NO) receptors. NO is a gas and does not bind to traditional receptors like GPCRs or nuclear. NO is a gas and does not bind to traditional receptors like GPCRs or nuclear receptors. Instead, it interacts with target proteins to mediate its effects. Examples. NO interacts with soluble guanylate cyclase (SGC) to activate the production of cyclic guanosine monophosphate (cGMP), which serves as a second messenger. NO also interacts with heme-containing proteins, such as hemoglobin and myoglobin, influencing their functions. In each case, these receptors facilitate the communication between hormones and their target cells, ultimately leading to physiological responses and regulation of various processes in the body. Regulation of receptors number in hormones in detail. Very long answer. ChatGPT. The regulation of receptor numbers is a crucial aspect of hormonal signaling, as it directly influences the responsiveness of target cells to hormones. Several mechanisms contribute to the regulation of receptor numbers, ensuring the appropriate cellular response to changing hormone levels. 1. Upregulation and downregulation. Upregulation. When hormone levels are low, cells may increase the number of receptors to enhance their sensitivity to the hormone. This is often observed in response to chronic low hormone levels. Downregulation. When hormone levels are high, cells may decrease the number of receptors to prevent overstimulation. This occurs in response to chronic high hormone levels. 2. Ligand-mediated regulation. Hormone receptor binding can trigger receptor internalization and degradation or recycling. This helps regulate receptor numbers and prevents desensitization. For example, GPCRs can undergo endocytosis upon hormone binding, leading to either receptor degradation or recycling back to the cell surface. 3. Feedback regulation. Negative feedback loops involve the hormone itself inhibiting its own release or signaling. For example, high levels of certain hormones, like insulin, can lead to a decrease in receptor numbers to prevent excessive cellular response. 4. Development. Hormone receptor binding can trigger receptor internalization and degradation or recycling. This helps regulate receptor numbers and prevents desensitization. For example, GPCRs can undergo endocytosis upon hormone binding, leading to either receptor degradation or recycling back to the cell surface. 3. Feedback regulation. Negative feedback loops involve the hormone itself inhibiting its own release or signaling. For example, high levels of certain hormones, like insulin, can lead to a decrease in receptor numbers to prevent excessive cellular response. 4. Developmental regulation. During development, receptor numbers may change as tissues and organs mature. For example, the growth hormone receptor increases in number during childhood, contributing to growth and development. 5. Disease and pathophysiological conditions. Certain diseases or conditions can lead to changes in receptor numbers. For instance, insulin resistance in type 2 diabetes can be associated with reduced insulin receptor numbers on target cells. 6. Hormonal regulation. Some hormones regulate the number of receptors for other hormones. For example, thyroid hormones regulate the expression of their own receptors. 7. Receptor desensitization and internalization. Prolonged exposure to high hormone levels can lead to receptor desensitization, where the cellular response diminishes even in the presence of the hormone. Internalization involves the endocytosis of receptors, leading to their sequestration from the cell surface. These mechanisms help maintain homeostasis by ensuring that cells respond appropriately to varying hormone levels. They enable cells to adapt to changing physiological conditions and prevent excessive activation or desensitization of hormone signaling pathways. Receptor activation of hormones in detail. Very long answer. ChatGPT. The activation of hormone receptors is a complex process that triggers a cascade of intracellular events leading to cellular responses. The mechanism of receptor activation varies based on the type of receptor and the signaling pathway it activates. Here, we'll discuss the general process of receptor activation for different types of hormone receptors. 1. G-protein coupled receptors (GPCRs). Hormone binding to GPCR induces a conformational change in the receptor. This change activates an associated G protein by exchanging GDP for GTP. The activated G protein dissociates into its subunits, G-alpha and GBG. G-alpha and GBG modulate various effectors, such as adenylate cyclase or phospholipase C, leading to the generation of intracellular messengers like CAMP or IP3. These messengers activate protein kinases, triggering a phosphorylation cascade that eventually leads to the cellular response. 2. Receptor tyrosine kinases, RTKs. Hormone binding to RTKs leads to receptor dimerization or oligomerization. This causes autophosphorylation of tyrosine residues within the receptor cytoplasmic domain. Phosphorylated tyrosine residues serve as docking sites for intracellular signaling molecules, including adaptors and kinases. These molecules activate multiple downstream pathways, such as the ROS-RAF-MAPK pathway or the PI3K-AKT pathway. Activation of these pathways leads to changes in gene expression, cell growth, survival, and other cellular responses. 3. Intracellular receptors, steroid hormones. Steroid hormones diffuse across the cell membrane and bind to intracellular receptors located in the cytoplasm or nucleus. Hormone binding triggers a conformational change in the receptor, exposing its DNA binding domain. The receptor hormone complex translocates to the nucleus and binds to specific DNA sequences called hormone response elements (HREs). This binding modulates gene transcription, leading to the synthesis of specific proteins that mediate the hormonal response. 4. Ligand-gated ion channels. Hormone binding to these receptors causes a conformational change that opens or closes an ion channel. The influx or efflux of ions, such as calcium or sodium, changes the membrane potential. This change in membrane potential triggers downstream signaling events or cellular responses. 5. Cytokine receptors. Cytokine receptors belong to the RTK superfamily and activate Janus kinase (JAK) and signal transducer and activator of transcription (STAT) pathways. Hormone binding to the receptor leads to receptor dimerization and activation of associated YOX. YOX phosphorylate tyrosine residues on the receptor and STAT proteins. Phosphorylated STATs dimerize, translocate to the nucleus, and modulate gene transcription. In summary, receptor activation is a dynamic process involving conformational changes, phosphorylation events, and the activation of intracellular pathways. The type of receptor dictates the specific signaling pathways activated and the cellular responses elicited. This diversity allows cells to respond to a wide range of hormonal cues and adapt to changing physiological conditions. Second messengers of hormone action in detail. Very long answer. ChatGPT. Second messengers play a crucial role in mediating the intracellular effects of hormone receptor interactions. They act as intermediaries that transmit signals from the hormone-bound receptor at the cell surface to the intracellular effectors, leading to various physiological responses. Here's an in-depth explanation of second messengers and their functions. 1. Cyclic adenosine monophosphate (CAMP). Produced by the enzyme adenylate cyclase upon activation by G-protein coupled receptors (GPCRs). CAMP activates protein kinase A (PKA) by binding to its regulatory subunits and releasing the catalytic subunits. Active PKA phosphorylates target proteins, affecting processes like gene expression, metabolism, and ion channel activity. Example: epinephrine binding to beta-adrenergic receptors in the liver triggers CAMP-mediated glycogen breakdown. 2. Inositol trisphosphate (IP3) and diacylglycerol (DAG). Generated by the hydrolysis of phosphatidylinositol 4,5-bisphosphate PIP2 in response to GPCR activation. IP3 triggers the release of calcium ions Ca2+ from the endoplasmic reticulum (ER). Ca2+ acts as a versatile intracellular messenger, affecting processes such as muscle contraction, secretion, and gene expression. DAG activates protein kinase C (PKC), which phosphorylates target proteins involved in cell growth, differentiation, and metabolism. Example: hormones like acetylcholine binding to muscarinic receptors in smooth muscle cells lead to IP3 and DAG-mediated contraction. 3. Calcium ions Ca2+. Released from intracellular stores or enters the cell through ion channels in response to hormone binding. Ca2+ can directly bind to proteins like calmodulin, which then activates enzymes like calmodulin-dependent protein kinase CAMK. Ca2+ calmodulin complexes can also modulate ion channel activity and gene expression. Example: parathyroid hormone (PTH) stimulates the release of Ca2+ from bone to maintain blood calcium levels. 4. Receptor activation of hormones in detail. Very long answer. ChatGPT. The activation of hormone receptors is a complex process that triggers a cascade of intracellular events leading to cellular responses. The mechanism of receptor activation varies based on the type of receptor and the signaling pathway it activates. Here, we'll discuss the general process of receptor activation for different types of hormone receptors. 1. G-protein coupled receptors (GPCRs). Hormone binding to GPCR induces a conformational change in the receptor. This change activates an associated G protein by exchanging GDP for GTP. The activated G protein dissociates into its subunits, G-alpha and GBG. G-alpha and GBG modulate various effectors, such as adenylate cyclase or phospholipase C, leading to the generation of intracellular messengers. G-alpha and GBG modulate various effectors. G-alpha and GBG modulate various effectors, such as adenylate cyclase or phospholipase C, leading to the generation of intracellular messengers like CAMP or IP3. These messengers activate protein kinases, triggering a phosphorylation cascade that eventually leads to the cellular response. 2. Receptor tyrosine kinases (RTKs). Hormone binding to RTKs leads to receptor dimerization or oligomerization. This causes autophosphorylation of tyrosine residues within the receptor cytoplasmic domain. Phosphorylated tyrosine residues serve as docking sites for intracellular signaling molecules, including adaptors and kinases. These molecules activate multiple downstream pathways, such as the ROS-RAF-MAPK pathway or the PI3K-AKT pathway. Activation of these pathways leads to changes in gene expression, cell growth, survival, and other cellular responses. 3. Intracellular receptors, steroid hormones. Steroid hormones diffuse across the cell membrane and bind to intracellular receptors located in the cytoplasm or nucleus. Hormone binding triggers a conformational change in the receptor, exposing its DNA binding domain. The receptor hormone complex translocates to the nucleus and binds to specific DNA sequences called hormone response elements (HREs). This binding modulates gene transcription, leading to the synthesis of specific proteins that mediate the hormonal response. 4. Ligand-gated ion channels. Hormone binding to these receptors causes a conformational change that opens or closes an ion channel. The influx or efflux of ions, such as calcium or sodium, changes the membrane potential. This change in membrane potential triggers downstream signaling events or cellular responses. 5. Cytokine receptors. Cytokine receptors belong to the RTK superfamily and activate Janus kinase (JAK) and signal transducer and activator of transcription (STAT) pathways. Hormone binding to the receptor leads to receptor dimerization and activation of associated YOX. YOX phosphorylate tyrosine residues on the receptor and STAT proteins. Phosphorylated STATs dimerize, translocate to the nucleus, and modulate gene transcription. In summary, receptor activation is a dynamic process involving conformational changes, phosphorylation events, and the activation of intracellular pathways. The type of receptor dictates the specific signaling pathways activated and the cellular responses elicited. This diversity allows cells to respond to a wide range of hormonal cues and adapt to changing physiological conditions. Second messengers of hormone action in detail. Very long answer. ChatGPT. Second messengers play a crucial role in mediating the intracellular effects of hormone receptor interactions. They act as intermediaries that transmit signals from the hormone-bound receptor at the cell surface to the intracellular effectors, leading to various physiological responses. Here's an in-depth explanation of second messengers and their functions. 1. Cyclic adenosine monophosphate (CAMP). Produced by the enzyme adenylate cyclase upon activation by G-protein coupled receptors (GPCRs). CAMP activates protein kinase A (PKA) by binding to its regulatory subunits and releasing the catalytic subunits. Active PKA phosphorylates target proteins, affecting processes like gene expression, metabolism, and ion channel activity. Example: epinephrine binding to beta-adrenergic receptors in the liver triggers CAMP-mediated glycogen breakdown. 2. Inositol trisphosphate (IP3) and diacylglycerol (DAG). Generated by the hydrolysis of phosphatidylinositol 4,5-bisphosphate PIP2 in response to GPCR activation. IP3 triggers the release of calcium ions Ca2+ from the endoplasmic reticulum (ER). Ca2+ acts as a versatile intracellular messenger, affecting processes such as muscle contraction, secretion, and gene expression. DAG activates protein kinase C (PKC), which phosphorylates target proteins involved in cell growth, differentiation, and metabolism. Example: hormones like acetylcholine binding to muscarinic receptors in smooth muscle cells lead to IP3 and DAG-mediated contraction. 3. Calcium ions, Ca2+. Released from intracellular stores or enters the cell through ion channels in response to hormone binding. Ca2+ can directly bind to proteins like calmodulin, which then activates enzymes like calmodulin-dependent protein kinase CAMK. Ca2+ calmodulin complexes can also modulate ion channel activity and gene expression. Example: parathyroid hormone (PTH) stimulates the release of Ca2+ from bone to maintain blood calcium levels. 4. Cyclic guanosine monophosphate (CGMP), produced by guanylate cyclase upon activation by hormones like nitric oxide (NO). CGMP activates protein kinase G(PKG), which phosphorylates target proteins involved in smooth muscle relaxation and other processes. Example: no release by endothelial cells triggers CGMP-mediated vasodilation. 5. Phosphoinositide 3 kinase PI3K pathway. 5. Phosphoinositide 3 kinase PI3K pathway. Activated by receptor tyrosine kinases and certain GPCRs. PI3K generates phosphatidylinositol 3, 4, 5-trisphosphate PIP3 from phosphatidylinositol 4, 5-bisphosphate PIP2. PIP3 recruits protein kinases like AKT, which phosphorylate various targets involved in cell survival, growth, and metabolism. Example: insulin binding to its receptor activates the PI3K pathway, promoting glucose uptake in cells. These second messengers amplify and relay the signal from hormone receptor interactions, allowing cells to respond to small amounts of hormone molecules. They ensure that the cellular response is precisely regulated and tailored to specific physiological needs. The complexity of second messenger signaling allows for a wide range of responses in different cell types and tissues, contributing to the diverse effects of hormones on various physiological processes. CAMP in hormones in detail. ChatGPT. Cyclic adenosine monophosphate (CAMP) is a critical second messenger that plays a central role in the intracellular signaling pathways of many hormones. It is synthesized from adenosine triphosphate (ATP) by the enzyme adenylate cyclase and acts as an intermediary between hormone receptor interactions at the cell surface and cellular responses in the cytoplasm and nucleus. Let's explore the role of CAMP in hormone action in detail. 1. Hormones that activate CAMP pathway. Several hormones bind to G-protein coupled receptors (GPCRs) on the cell surface, triggering the activation of adenylate cyclase. Adenylate cyclase catalyzes the conversion of ATP into CAMP. Notable examples of hormones that use the CAMP pathway include epinephrine and norepinephrine, catecholamines, glucagon. Adrenocorticotropic hormone (ACTH), thyroid-stimulating hormone (TSH), parathyroid hormone (PTH), vasopressin. 2. CAMP production and degradation. Upon binding of these hormones to the respective receptors, G proteins are activated. G proteins, in turn, stimulate adenylate cyclase to produce CAMP. CAMP acts as a second messenger by binding to an activating protein kinase APKA. The action of CAMP is short-lived because it is rapidly degraded by the enzyme phosphodiesterase PDE back to AMP. 3. Activation of protein kinase APKA. CAMP binds to the regulatory subunits of PKA, causing their dissociation from the catalytic subunits. The free catalytic subunits are then active and capable of phosphorylating various target proteins. PKA targets include enzymes, ion channels, and transcription factors. Phosphorylation by PKA leads to a cascade of intracellular events that ultimately produce a physiological response to the hormone. 4. Cellular responses to CAMP signaling. The CAMP pathway plays a central role in regulating a wide range of cellular processes. Metabolism. CAMP signaling stimulates glycogen breakdown, glycogenolysis, and inhibits glycogen synthesis, glycogenesis, leading to increased glucose availability. Ion channels. CAMP-mediated phosphorylation affects ion channel activity, influencing membrane potential and cellular excitability. Gene expression. PKA can enter the nucleus and phosphorylate transcription factors, modulating gene expression. This has implications for cell growth, differentiation, and hormone secretion. CAMP stimulates the release of hormones from various endocrine glands, such as glucagon from the pancreas. 5. Physiological implications. The CAMP pathway allows for rapid and specific responses to hormonal signals. Different cells can respond differently to the same hormone depending on their complement of receptors and downstream effectors. The CAMP pathway's versatility makes it crucial for coordinating physiological processes, ranging from metabolic regulation to neurotransmitter release. In summary, CAMP serves as a universal secondary messenger that relays extracellular hormonal signals to intracellular responses. The CAMP pathway is central to the actions of various hormones and contributes to the diverse effects they elicit across different tissues and physiological contexts. CGMP in hormones in detail. Very long answer. ChatGPT. Cyclic guanosine monophosphate (CGMP) is another important second messenger that plays a vital role in the intracellular signaling pathways of various hormones. It is synthesized from guanosine triphosphate (GTP) by the enzyme guanylate cyclase and serves as a mediator between extracellular hormone receptor interactions and cellular responses. Here's a detailed explanation of the role of CGMP in hormone action. 1. Hormones that activate CGMP pathway. Similar to CAMP, CGMP is activated by hormones that bind to G-protein coupled receptors (GPCRs) on the cell surface. These hormones stimulate guanylate cyclase, which converts GTP into CGMP. Examples of hormones that utilize the CGMP pathway include nitric oxide (NO), natriuretic peptides, atrial natriuretic peptide, and brain natriuretic peptide. 2. CGMP production and degradation. Nitric oxide (NO), natriuretic peptides, atrial natriuretic peptide, and brain natriuretic peptide. 2. CGMP production and degradation. Upon hormone receptor interaction, G proteins are activated, leading to the stimulation of guanylate cyclase. Guanylate cyclase produces CGMP from GTP. Unlike CAMP, CGMP is not degraded by phosphodiesterase PDE, but rather by a family of enzymes known as phosphodiesterase 5-PDE5 or phosphodiesterase 6-PDE6 depending on the cell type. 3. Activation of protein kinase G-PKG. Once CGMP is produced, it binds to and activates protein kinase G-PKG. Like protein kinase APKA in the CAMP pathway, PKG is a serine threonine kinase that phosphorylates specific target proteins. Activation of PKG initiates downstream signaling events that modulate cellular responses. 4. Cellular responses to CGMP signaling. The CGMP pathway controls various physiological processes in cells. Vascular smooth muscle relaxation. One of the most well-known roles of CGMP is its involvement in vasodilation. CGMP stimulates the relaxation of smooth muscle cells in blood vessel walls, leading to increased blood flow. Sodium excretion. Natriuretic peptides stimulate CGMP production, which promotes sodium excretion by the kidneys, contributing to blood pressure regulation. Visual signal transduction. In photoreceptor cells of the retina, CGMP plays a role in the regulation of ion channels in the process of visual signal transduction. 5. Physiological implications. The CGMP pathway is crucial for maintaining cardiovascular homeostasis, blood pressure regulation, and other physiological functions. Dysregulation of CGMP signaling can lead to various cardiovascular diseases, including hypertension and heart failure. In summary, CGMP serves as a second messenger that relays hormonal signals to intracellular responses, particularly in the context of vasodilation, sodium excretion, and visual signal transduction. Its role in various physiological processes underscores its importance in maintaining normal cellular function and overall health. Inositol triphosphate in hormones in detail. Very long answer. ChatGPT. Inositol triphosphate (IP3) is a crucial intracellular second messenger that plays a significant role in hormone signaling pathways. It is derived from the membrane phospholipid phosphatidylinositol 4, 5-bisphosphate PIP2, and is involved in regulating a wide range of cellular processes. Here's a detailed explanation of the role of IP3 in hormone action. 1. Hormones that activate IP3 pathway. IP3 is activated in response to hormone receptor interactions that involve G-protein coupled receptors (GPCRs) or receptor tyrosine kinases (RTKs). Hormones such as acetylcholine, epinephrine, and various growth factors can activate IP3 pathways. 2. Activation of phospholipase C (PLC). Hormone binding to its receptor triggers the activation of phospholipase C (PLC), an enzyme associated with the cell membrane. PLC cleaves phosphatidylinositol 4, 5-bisphosphate KIP2 into 2-second messengers, diacylglycerol DAG, and inositol triphosphate IP3. 3. IP3 binding to receptors on endoplasmic reticulum ER. IP3 diffuses into the cytoplasm and binds to specific receptors located on the membrane of the endoplasmic reticulum ER. These IP3 receptors are calcium channels that, when activated by IP3 binding, allow the release of calcium ions Ca2+ from the urine into the cytoplasm. 4. Calcium release and intracellular signaling. The release of Ca2+ from the urine into the cytoplasm is a key event triggered by IP3. The increased cytosolic Ca2+ concentration serves as a secondary messenger that regulates various cellular processes. It can bind to and activate specific proteins, including calmodulin, which further propagate downstream signaling pathways. 5. Physiological implications. IP3-mediated Ca2+ signaling has diverse physiological implications. Muscle contraction. In muscle cells, IP3-induced Ca2+ release plays a role in muscle contraction. Secretion. In exocrine cells, IP3-mediated Ca2+ release is essential for the secretion of various substances, such as neurotransmitters and hormones. Cell proliferation and differentiation. IP3-mediated signaling contributes to cell proliferation and differentiation in response to growth factors. 6. Termination of IP3 signaling. The IP3 pathway is tightly regulated to prevent sustained signaling. IP3 is rapidly metabolized by inositol polyphosphate phosphatase, IPASIS, converting it back to inositol monophosphate. Additionally, Ca2+ pumps in the ER membrane actively transport Ca2+ back into the ER, reducing cytoplasmic Ca2+ levels. 7. Pharmacological relevance. IP3-mediated signaling is a target for drug development. For instance, certain pharmaceutical agents target the IP3 receptor to modulate intracellular Ca2+ levels and treat conditions like cardiovascular diseases and neurodegenerative disorders. In summary, inositol triphosphate IP3 is a vital intracellular second messenger that is generated in response to hormone receptor interactions. It triggers the release of Ca2+ from the ER, leading to diverse cellular responses, including muscle contraction, secretion, and cell proliferation. The tight regulation of IP3 signaling ensures proper cellular function and physiological homeostasis. Diacylglycerol in hormones in detail. Very long answer. ChatGPT. Diacylglycerol DAG is another important intracellular second messenger involved in hormone signaling pathways. It is derived from the cleavage of phosphatidylinositol 4, 5-bisphosphate KIP2 by the enzyme phospholipase C (PLC). DAG, along with inositol triphosphate IP3, plays a key role in transducing hormonal signals to regulate various cellular processes. Here's a detailed explanation of the role of DAG in hormone action. 1. Hormones that activate DAG pathway. DAG is activated in response to hormone receptor interactions that involve G-protein coupled receptors (GPCRs) or receptor tyrosine kinases (RTKs). Hormones such as epinephrine, acetylcholine, and various growth factors can activate DAG pathways. 2. Activation of phospholipase C (PLC). Hormone binding to its receptor triggers the activation of phospholipase C (PLC), an enzyme associated with the cell membrane. PLC cleaves phosphatidylinositol 4, 5-bisphosphate KIP2 into 2-second messengers, diacylglycerol DAG, and inositol triphosphate IP3. 3. DAG's role in protein kinase C (PKC) activation. DAG is an activator of protein kinase C (PKC), a family of serine threonine kinases. PKC is recruited to the cell membrane by DAG, where it becomes activated in the presence of both DAG and Ca2+ ions. Activated PKC phosphorylates specific target proteins, thereby transmitting the hormonal signal and initiating various cellular responses. 4. Physiological implications. DAG-mediated PKC signaling has diverse physiological implications. Cellular proliferation. DAG-mediated PKC activation is involved in cell growth and proliferation, which are crucial for tissue development and repair. Neuronal signaling. In neurons, DAG-mediated PKC activation modulates synaptic plasticity and neurotransmitter. Neuronal signaling. In neurons, DAG-mediated PKC activation modulates synaptic plasticity and neurotransmitter release. Immune response. DAG-PKC signaling plays a role in immune responses, including the activation of immune cells and inflammatory processes. 5. Termination of DAG signaling. The DAG-PKC pathway is tightly regulated to prevent excessive signaling. DAG levels are controlled by the metabolism of phosphatidylinositol 4, 5-bisphosphate KIP2 by PLC. Additionally, DAG can be converted back to phosphatidic acid through the action of DAG kinases, which terminates PKC activation. 6. Pharmacological relevance. The DAG-PKC pathway is a target for drug development. Drugs that modulate PKC activity are being explored for various therapeutic applications, including cancer treatment, cardiovascular diseases, and neurological disorders. In summary, diacylglycerol DAG is an important intracellular second messenger generated in response to hormone receptor interactions. It activates protein kinase C (PKC), which in turn initiates a cascade of signaling events leading to diverse cellular responses. The DAG-PKC pathway is involved in cell proliferation, neuronal signaling, and immune responses, and its tight regulation ensures proper cellular function and homeostasis. Receptor signal transduction in hormones in detail. Very long answer. ChatGPT. Receptor signal transduction in hormone action is a complex process by which hormones transmit their signals from the extracellular environment to the intracellular space, leading to a wide range of physiological responses. Here's a detailed explanation of the receptor signal transduction process. 1. Hormone receptor interaction. The process begins with the binding of a hormone to its specific receptor. Hormone receptors can be membrane bound, such as G-protein coupled receptors (GPCRs) or receptor tyrosine kinases (RTKs), or intracellular, like nuclear receptors. 2. G-protein coupled receptors (GPCRs). In GPCRs, hormone binding induces a conformational change in the receptor, which activates a nearby G-protein. The G-protein consists of 3 subunits: alpha, beta, and gamma. Upon activation, the alpha subunit dissociates from the beta-gamma subunits and can activate intracellular signaling pathways. 3. Activation of G-proteins. Activated G-proteins can stimulate or inhibit the activity of effector molecules such as adenylate cyclase or phospholipase C (PLC), leading to the generation of second messengers like cyclic AMP (CAMP) or inositol triphosphate IP3 and diacylglycerol DAG. 4. Receptor tyrosine kinases (RTKs). RTKs are transmembrane proteins with intrinsic kinase activity. Upon hormone binding, RTKs dimerize and undergo autophosphorylation, activating their kinase domains. This triggers a phosphorylation cascade involving downstream signaling molecules, including adaptor proteins and enzymes like ROS. 5. Activation of second messengers. Second messengers such as CAMP, IP3, and DAG are crucial in transmitting hormone signals. They modulate the activity of various protein kinases, including protein kinase A (PKA) for CAMP and protein kinase C (PKC) for DAG and IP3, leading to the phosphorylation of target proteins. 6. Activation of protein kinases. Phosphorylated protein kinases, like PKA, PKC, and mitogen-activated protein kinases (MAPKs) phosphorylate target proteins involved in various cellular functions, including gene expression, metabolism, cell growth, and differentiation. 7. Nuclear receptors. Some hormones, like steroid hormones, diffuse across the cell membrane and bind to nuclear receptors in the cytoplasm. The hormone receptor complex translocates to the nucleus, where it binds to specific DNA sequences, hormone response elements, and regulates gene transcription. 8. Crosstalk and integration. Different signaling pathways can crosstalk and integrate, allowing the cell to respond to multiple stimuli simultaneously. This provides specificity and flexibility in the cellular response to hormones. 9. Negative feedback. To maintain homeostasis, hormonal signaling often involves negative feedback loops. When the desired cellular response is achieved, negative feedback mechanisms are activated to dampen the signaling cascade and prevent overstimulation. 10. Duration of signaling. Hormone receptor interactions can be transient or prolonged, depending on the receptor type and the nature of the signal. GPCR signaling tends to be faster and transient, while nuclear receptor signaling may lead to longer-lasting changes in gene expression. In summary, receptor signal transduction in hormone action involves a complex series of events, including hormone receptor interactions, activation of GPCRs or RTKs, generation of second messengers, activation of protein kinases, and modulation of gene expression. This intricate process allows cells to respond to hormones in a highly regulated and specific manner, ultimately coordinating various physiological responses in the body. Process steps in detail. ChatGPT. Certainly, let's break down the process of receptor signal transduction in hormone action into more detailed steps. 1. Hormone binding to receptor. The process begins when a hormone ligand binds to its specific receptor on the cell surface or within the cell. The binding induces a conformational change in the receptor, activating it. 2. Activation of G protein coupled receptors, GPCRs. In GPCRs, the activated receptor interacts with a G protein, which consists of alpha, beta, and gamma subunits. The alpha subunit of the G protein exchanges GDP for GTP and dissociates from the beta gamma subunits. 3. Activation of effector molecules. Activated G alpha subunit or other signaling proteins, like ROS, stimulate downstream effector molecules such as adenylate cyclase or phospholipase C PLC. Adenylate cyclase produces cyclic AMP camp, while PLC cleaves phosphatidylanosytol 4, 5-bisphosphate PIP2 into inositol trisphosphate IP3 and diacylglycerol DAG. 4. Second messenger generation. CAMP, IP3, and DAG are second messengers that act as intracellular signaling molecules. CAMP activates protein kinase A PKA, while IP3 binds to receptors on the endoplasmic reticulum, releasing Ca2+ ions into the cytoplasm. DAG activates protein kinase C PKC. 5. Protein kinase activation. PKA, PKC, and other protein kinases are activated upon binding of CAMP, Ca2+, or DAG. These kinases phosphorylate specific target proteins, including enzymes, transcription factors, and ion channels. 6. Amplification of signal. A single hormone receptor interaction can lead to the activation of multiple G proteins, resulting in the amplification of the signal. This ensures that even a small number of hormone molecules can have a significant physiological effect. 7. Translocation to nuclear receptors. Some hormones, like steroid hormones, diffuse across the cell membrane and bind to nuclear receptors in the cytoplasm. The hormone receptor complex translocates to the nucleus, where it binds to hormone response elements on DNA, regulating gene transcription. 8. Integration of signaling pathways. Different receptors can activate distinct signaling pathways in the same cell. The cell integrates these signals to produce a coordinated response that reflects the overall physiological context. 9. Negative feedback and termination. Negative feedback loops are activated to prevent overstimulation. This may involve the downregulation of hormone receptors, dephosphorylation of signaling proteins, or internalization of receptor ligand complexes. 10. Cellular response. The phosphorylation of target proteins by protein kinases modifies their activity, leading to changes in cell function, metabolism, gene expression, and cell growth. Ultimately, these changes mediate the specific physiological response to the hormone. 11. Removal of hormone signal. The hormone signal is terminated by receptor internalization, degradation of second messengers, and dephosphorylation of target proteins. This ensures that the cell's responsiveness to subsequent hormone signals is restored. In summary, the process of receptor signal transduction in hormone action involves a series of highly coordinated steps, from hormone receptor interaction to activation of second messengers, protein kinases, and downstream effectors. This process allows cells to convert extracellular hormonal signals into specific intracellular responses, influencing various physiological functions in the body. Isosinoids and hormone action in detail. Very long answer. ChatGPT. Eicosanoids are a group of bioactive lipid molecules derived from arachidonic acid, a polyunsaturated fatty acid present in cell membranes. These molecules play crucial roles in mediating various physiological processes and are intimately involved in hormone action. Let's delve into the details. 1. Eicosanoid biosynthesis. Eicosanoids are synthesized from arachidonic acid through enzymatic reactions involving cyclooxygenases, COX, lipoxygenases, LOX, and cytochrome P450 enzymes. The three main classes of eicosanoids are prostaglandins, thromboxanes, and leukotrienes. 2. Prostaglandins and thromboxanes. Prostaglandins PGs and thromboxanes TXs are synthesized from arachidonic acid via the COX pathway. COX enzymes COX1 and COX2 convert arachidonic acid into PGH2, which is then converted into various PGs or TXs by tissue-specific enzymes. Prostaglandins regulate numerous physiological processes, including inflammation, pain, fever, blood vessel dilation, and uterine contractions. Thromboxanes are involved in platelet aggregation and vasoconstriction. 3. Leukotrienes. Eicosanoids are synthesized from arachidonic acid through Eicosanoids are a group of bioactive lipid molecules derived from arachidonic acid, a polyunsaturated fatty acid present in cell membranes. These molecules play crucial roles in mediating various physiological processes and are intimately involved in hormone action. Let's delve into the details. 1. Eicosanoid biosynthesis. Eicosanoids are synthesized from arachidonic acid through enzymatic reactions involving cyclooxygenases, COX, lipoxygenases, LOX, and cytochrome. Eicosanoids are synthesized from arachidonic acid through enzymatic reactions involving cyclooxygenases, COX, lipoxygenases, LOX, and cytochrome P450 enzymes. The three main classes of eicosanoids are prostaglandins, thromboxanes, and leukotrienes. 2. Prostaglandins and thromboxanes. Prostaglandins PGs and thromboxanes TXs are synthesized from arachidonic acid via the COX pathway. COX enzymes COX1 and COX2 convert arachidonic acid into PGH2, which is then converted into various PGs or TXs by tissue-specific enzymes. Prostaglandins regulate numerous 2. Prostaglandins and thromboxanes. Prostaglandins PGs and thromboxanes TXs are synthesized from arachidonic acid via the COX pathway. COX enzymes COX1 and COX2 convert arachidonic acid into PGH2, which is then converted into various PGs or TXs by tissue-specific enzymes. Prostaglandins regulate numerous physiological processes, including inflammation, pain, fever, blood vessel dilation, and uterine contractions. Thromboxanes are involved in platelet aggregation and vasoconstriction. 3. Leukotrienes. Leukotrienes are synthesized from arachidonic acid via the LOX pathway. They are involved in immune responses, particularly in allergic reactions and inflammation. Leukotrienes mediate bronchoconstriction and contribute to asthma symptoms. 4. Hormone interaction with eicosanoids. COX enzymes COX1 and prostaglandins PGs and thromboxanes TXs are synthesized from arachidonic acid via the COX pathway. COX enzymes COX1 and COX2 convert arachidonic acid into PGH2, which is then converted into various PGs or TXs by tissue-specific enzymes. Prostaglandins regulate numerous physiological processes, including inflammation, pain, fever, blood vessel dilation, and uterine contractions. Thromboxanes are involved in platelet aggregation and vasoconstriction. 3. Leukotrienes. Leukotrienes are synthesized from arachidonic acid via the LOX pathway. They are involved in immune responses, particularly in allergic reactions and inflammation. Leukotrienes mediate bronchoconstriction and contribute to asthma symptoms. 4. Hormone interaction with eicosanoids. Hormones can stimulate or modulate the synthesis of eicosanoids in response to various stimuli. For example, during inflammation, hormones such as glucocorticoids can inhibit COX2 expression, thereby reducing the production of pro-inflammatory prostaglandins. 5. Interaction with hormone receptors. Eicosanoids can bind to specific G protein coupled receptors on the surface of target cells. The receptors for eicosanoids are part of the rhodopsin-like GPCR family. The binding of eicosanoids to the receptors triggers intracellular signaling pathways, resulting in various cellular responses. 6. Regulation of immune responses. Eicosanoids, especially leukotrienes and prostaglandins, play a critical role in regulating immune responses. They attract immune cells to sites of infection or injury and contribute to the inflammatory response. 7. Hormonal regulation of eicosanoid synthesis. Some hormones, like glucocorticoids, can modulate the synthesis of eicosanoids. Glucocorticoids inhibit phospholipase A2, an enzyme that releases arachidonic acid from cell membranes, thereby reducing the availability of substrate for eicosanoid synthesis. This anti-inflammatory action of glucocorticoids contributes to the therapeutic effects. 8. Cellular responses to eicosanoids. The cellular responses to eicosanoids are diverse and context-dependent. Prostaglandins can induce vasodilation, increase vascular permeability, regulate blood pressure, and influence pain sensation. Thromboxanes promote platelet aggregation and vasoconstriction. Leukotrienes contribute to bronchoconstriction, vascular permeability, and inflammation. 9. Interplay with other signaling pathways. Eicosanoid signaling pathways often interact with other signaling pathways, such as those activated by growth factors and cytokines. These interactions. 8. Cellular responses to eicosanoids. The cellular responses to eicosanoids are diverse and context-dependent. Prostaglandins can induce vasodilation, increase vascular permeability, regulate blood pressure, and influence pain sensation. Thromboxanes promote platelet aggregation and vasoconstriction. Leukotrienes contribute to bronchoconstriction, vascular permeability, and inflammation. 9. Interplay with other signaling pathways. Eicosanoid signaling pathways often interact with other signaling pathways, such as those activated by growth factors and cytokines. These interactions can amplify or modulate the overall cellular response. In summary, eicosanoids are versatile lipid molecules derived from arachidonic acid and play integral roles in mediating various physiological processes. Their interactions with hormone signaling pathways contribute to the regulation of immune responses, inflammation, blood vessel tone, and other vital functions in the body. The complex interplay between hormones. Point. ChatGPT. Certainly, here are more points about the interaction between eicosanoids and hormone action. 10. Hormonal modulation of eicosanoid synthesis. Hormones like estrogen and progesterone can influence the synthesis of eicosanoids. Estrogen may enhance the production of certain prostaglandins, impacting reproductive processes. Progesterone can suppress the production of prostaglandins and leukotrienes, playing a role in pregnancy maintenance. 11. Crosstalk with hormone signaling. Eicosanoids can modulate the effects of other hormones and vice versa. For example, some eicosanoids can impact insulin sensitivity and glucose metabolism. Insulin can influence eicosanoid production, affecting inflammation and lipid metabolism. 12. Role in reproductive physiology. Eicosanoids play crucial roles in reproductive processes. Prostaglandins are involved in ovulation, implantation, and uterine contractions during labor. Prostaglandins also play a role in regulating the menstrual cycle. 13. Role in cardiovascular health. Prostaglandins and thromboxanes influence vascular tone and blood pressure regulation. Some prostaglandins promote vasodilation and maintain blood vessel health. Others, like thromboxane A2, contribute to vasoconstriction and platelet aggregation. 14. Inflammatory and autoimmune disorders. Dysregulation of eicosanoid production is associated with various inflammatory and autoimmune disorders. For example, excessive production of prostaglandins can contribute to chronic inflammation and pain in conditions like rheumatoid arthritis. 15. Therapeutic implications. Eicosanoids are targets for drug development. Nonsteroidal anti-inflammatory drugs (NSAIDs) inhibit COX enzymes to reduce pain and inflammation. Aspirin irreversibly inhibits COX-1 and decreases platelet aggregation. 16. Dietary omega-3 fatty acids. Omega-3 fatty acids, found in fish oil, can be converted into anti-inflammatory eicosanoids. These eicosanoids compete with pro-inflammatory eicosanoids, potentially reducing inflammation. 17. Cellular specificity. Eicosanoids can have diverse effects depending on the cell type and receptor expression. Different tissues respond differently to the same eicosanoid. 18. Hormonal regulation of enzymes. Hormones can regulate the expression of enzymes involved in eicosanoid synthesis. For instance, insulin can influence the activity of enzymes that release arachidonic acid from cell membranes. 19. Impact on nervous system. Eicosanoids are involved in synaptic signaling and neurotransmission in the nervous system. They influence the release of neurotransmitters and regulate neuronal excitability. 20. Role in immunity. Eicosanoids regulate immune responses and inflammation. They attract immune cells to sites of infection or injury and help coordinate immune reactions. In conclusion, the interactions between eicosanoids and hormone action are intricate and multifaceted. Eicosanoids, derived from arachidonic acid, play vital roles in modulating physiological responses, regulating inflammation, and maintaining homeostasis. Their interactions with hormonal signaling pathways have profound effects on various bodily functions, making them important targets for research and potential therapeutic interventions. Anatomy of endocrine glands in detail. Very long answer. ChatGPT. Certainly, here's a detailed overview of the anatomy of some major endocrine glands. 1. Pituitary gland hypothesis. The pituitary gland is located at the base of the brain in a bony cavity called the sella turcica. It consists of two main parts: the anterior pituitary adenohypophysis. The pituitary gland is located at the base of the brain in a bony cavity called the sella turcica. It consists of two main parts: the anterior pituitary adenohypophysis. It consists of two main parts: the anterior pituitary adenohypophysis and the posterior pituitary neurohypophysis. The anterior pituitary is composed of glandular tissue, while the posterior pituitary is an extension of neural tissue. 2. Thyroid gland. The thyroid gland is situated in the neck, just below the Adam's apple. It consists of two lobes connected by an isthmus. The thyroid gland is composed of follicles that produce thyroid hormones, primarily thyroxine T4 and triiodothyronine T3. 3. Parathyroid glands. There are typically 4 parathyroid glands, 2 on each side, located near the thyroid gland. These small glands are responsible for regulating calcium and phosphate levels in the blood through the secretion of parathyroid hormone PTH. 4. Adrenal glands. There are 2 adrenal glands, one on top of each kidney. Each adrenal gland has 2 distinct parts: the outer cortex and the inner medulla. The adrenal cortex produces steroid hormones such as cortisol, aldosterone, and sex hormones. The adrenal medulla secretes catecholamines like adrenaline and noradrenaline. 5. Pancreas. The pancreas is located behind the stomach. It has both endocrine and exocrine functions. The endocrine portion consists of clusters of cells called the islets of Langerhans, which produce hormones such as insulin and glucagon. 6. Pineal gland. The pineal gland is a small gland located deep within the brain, attached to the roof of the third ventricle. It produces the hormone melatonin, which plays a role in regulating circadian rhythms and sleep-wake cycles. 7. Gonads, ovaries, and testes. Ovaries are located in the pelvic cavity in females and produce estrogen and progesterone. Testes are located in the scrotum in males and produce testosterone and sperm. 8. Thymus. The thymus is located in the upper chest, behind the sternum. It plays a role in the development of T lymphocytes, T cells important for immune responses. 9. Hypothalamus. While not a gland in the traditional sense, the hypothalamus is a crucial part of the endocrine system. It is located in the brain and controls the pituitary gland by releasing hormones that stimulate or inhibit its functions. Each endocrine gland has a unique structure tailored to its specific functions. These glands collectively regulate various physiological processes, including growth, metabolism, stress response, reproduction, and more. The intricate connections and interactions between these glands ensure proper communication and coordination of the body's hormonal responses. Structure of endocrine glands in detail. Very long answer. ChatGPT. Certainly, here's a detailed overview of the structure of some major endocrine glands. 1. Pituitary gland hypothesis. The pituitary gland is divided into 2 parts: the anterior pituitary adenohypophysis and the posterior pituitary neurohypophysis. Anterior pituitary, composed of glandular tissue with various cell types, including somatotropes produce growth hormone, corticotropes produce adrenocorticotropic. The pituitary gland is divided into two parts, the anterior pituitary adenohypophysis and the posterior pituitary neurohypophysis. The pituitary gland is divided into two parts, the anterior pituitary adenohypophysis and the posterior pituitary neurohypophysis. Anterior pituitary, composed of glandular tissue with various cell types, including somatotropes produce growth hormone, corticotropes produce adrenocorticotropic hormone, gonadotropes produce luteinizing hormone and follicle-stimulating hormone, thyrotropes produce thyroid-stimulating hormone, and lactotropes produce prolactin. Posterior pituitary, composed of neural tissue and stores and releases hormones produced by the hypothalamus, such as oxytocin and vasopressin. 2. Thyroid gland. The thyroid gland consists of two lobes connected by an isthmus. Each lobe is made up of many follicles, which are spherical structures containing colloid thyroglobulin. The follicular cells lining the follicles produce thyroid hormones T3 and T4, which are stored in the colloid. 3. Parathyroid glands. Typically 4 parathyroid glands located on or embedded in the posterior surface of the thyroid gland. Each gland is composed of chief cells that produce parathyroid hormone PTH and oxycyl cells of uncertain function. 4. Adrenal glands. Each adrenal gland has an outer cortex and an inner medulla. 2. Thyroid gland. The thyroid gland consists of two lobes connected by an isthmus. Each lobe is made up of many follicles, which are spherical structures containing colloid thyroglobulin. The follicular cells lining the follicles produce thyroid hormones T3 and T4, which are stored in the colloid. 3. Parathyroid glands. Typically 4 parathyroid glands located on or embedded in the posterior surface of the thyroid gland. Each gland is composed of chief cells that produce parathyroid hormone PTH and oxycyl cells of uncertain function. 4. Adrenal glands. Each adrenal gland has an outer cortex and an inner medulla. Adrenal cortex, divided into 3 zones. Zona glomerulosa produces aldosterone, zona fasciculata produces cortisol, and zona reticularis produces androgens. Adrenal medulla produces catecholamines, including adrenaline, epinephrine, and noradrenaline, norepinephrine. 5. Pancreas. The pancreas has both endocrine and exocrine functions. The endocrine portion consists of clusters of cells called islets of Langerhans. The islets contain different cell types. Alpha cells secrete glucagon, beta cells secrete insulin, delta cells secrete somatostatin, and PP cells secrete pancreatic polypeptide. 6. Pineal. The islets contain different cell types. Alpha cells secrete glucagon, beta cells secrete insulin, delta cells secrete somatostatin, and PP cells secrete pancreatic polypeptide. 6. Pineal gland. The pineal gland is a small, pinecone-shaped structure in the brain. It is composed of pinealocytes and glial cells. Pinealocytes secrete melatonin, a hormone involved in regulating circadian rhythms. 7. Gonads, ovaries, and testes. Ovaries, composed of ovarian follicles containing oocytes and granulosa cells that produce estrogen and progesterone. Testes, composed of seminiferous tubules where spermatogenesis occurs. Interstitial cells, Leydig cells produce testosterone. 8. Thymus. The thymus is located in the upper chest behind the sternum. It consists of lobules containing a cortex and a medulla. The cortex contains immature T cells, while the medulla contains mature T cells. 9. Hypothalamus. While not a gland, the hypothalamus is crucial in controlling the endocrine system. It contains various nuclei that produce releasing and inhibiting hormones, which regulate the anterior pituitary's hormone release. Each endocrine gland structure is tailored to its specific functions, allowing them to produce and secrete hormones that play essential roles in maintaining homeostasis, growth, reproduction, metabolism, and more in the body. Physiological functions. Endocrine glands in detail. Very long answer. ChatGPT. Certainly, here's a detailed overview of the physiological functions of some major endocrine glands. 1. Pituitary gland hypothesis. Anterior pituitary produces and releases several hormones that regulate other endocrine glands, such as growth hormone GH, adrenocorticotropic hormone ACTH, thyroid-stimulating hormone TSH, luteinizing hormone LH, follicle-stimulating hormone FSH, and prolactin. Posterior pituitary stores and releases hormones produced by the hypothalamus, including oxytocin, uterine contractions during labor and milk ejection, and vasopressin regulates water balance. Anterior pituitary produces and releases several hormones that regulate other endocrine glands, such as growth hormone GH, adrenocorticotropic hormone ACTH, thyroid-stimulating hormone TSH, luteinizing hormone LH, follicle-stimulating hormone FSH, and prolactin. Posterior pituitary stores and releases hormones produced by the hypothalamus, including oxytocin, uterine contractions during labor and milk ejection, and vasopressin regulates water balance. 2. Thyroid gland produces thyroid hormones T3 and T4, which regulate metabolism, growth, and development. Produces calcitonin, which regulates calcium levels in the blood by promoting its deposition in bone. 3. Parathyroid glands produces parathyroid hormone PTH, which regulates calcium and phosphate levels in the blood by increasing calcium absorption from the intestines, reducing calcium excretion by the kidneys, and releasing calcium from bone. 4. Adrenal glands. Adrenal cortex produces hormones essential for maintaining fluid balance, aldosterone, regulating metabolism and stress response, cortisol, and influencing secondary sexual characteristics, androgens. Adrenal medulla produces adrenaline, epinephrine, and noradrenaline, norepinephrine, which are involved in the fight or flight response. 5. Pancreas. Islets of Langerhans produce hormones that regulate blood glucose levels. Insulin lowers blood glucose by promoting its uptake by cells, and glucagon raises blood glucose by promoting its release from storage. 6. Pineal gland secretes melatonin, which helps regulate sleep-wake cycles, circadian rhythms, and influences seasonal behaviors in some animals. 7. Gonads, ovaries, and testes. Ovaries produce estrogen and progesterone, which regulate the menstrual cycle, support pregnancy, and develop female secondary sexual characteristics. Testes produce testosterone, which regulates sperm production and male secondary sexual characteristics. 8. Thymus plays a vital role in the development of T lymphocytes, T cells, which are crucial for the immune system's proper functioning. 9. Hypothalamus produces hormones that regulate the anterior pituitary's hormone release, including thyrotropin-releasing hormone (TRH), corticotropin-releasing hormone (CRH), and gonadotropin-releasing hormone (GnRH). Each endocrine gland's physiological functions contribute to maintaining homeostasis, regulating growth and development, responding to stress, supporting reproductive functions, influencing metabolism, and ensuring proper functioning of various bodily systems. The complex interplay of these glands and their hormones orchestrates a multitude of physiological processes in the body. Control of secretion of hormones of endocrine glands in detail. Very long answer. ChatGPT. Certainly, let's delve into the detailed control of hormone secretion in various endocrine glands. 1. Pituitary gland. Hypothalamus pituitary axis. The hypothalamus releases releasing hormones e.g., TRH, CRH, GnRH that stimulate or inhibit the anterior pituitary to release or suppress hormone secretion. The hormones include TSH, ACTH, LH, FSH, GH, and prolactin. Feedback loops. The feedback from peripheral target organs or downstream hormones regulates hormone secretion. For instance, thyroid hormones inhibit TRH and TSH release when their levels are sufficient. 2. Thyroid gland. Hypothalamus pituitary thyroid axis. The hypothalamus releases TRH, which stimulates the anterior pituitary to release TSH. TSH, in turn, stimulates the thyroid gland to produce and release T3 and T4. Negative feedback. Elevated T3 and T4 levels inhibit TRH and TSH release, maintaining homeostasis. 3. Parathyroid glands. Calcium sensing. Specialized calcium sensing receptors in parathyroid cells regulate PTH secretion. Low blood calcium levels stimulate PTH release, increasing calcium levels. 4. Adrenal glands. Hypothalamus pituitary adrenal axis. In response to stress, the hypothalamus releases CRH, which stimulates the anterior pituitary to release ACTH. ACTH stimulates the adrenal cortex to secrete cortisol and aldosterone. Renin-angiotensin-aldosterone system. ROS. Low blood pressure or sodium levels stimulate renin release by the kidneys, leading to aldosterone secretion. 5. Pancreas. Glucose regulation. Elevated blood glucose stimulates insulin release from pancreatic beta cells, promoting glucose uptake. Low blood glucose stimulates glucagon release from alpha cells, which increases glucose production. 6. Pineal gland. Light-dark cycle. The pineal gland responds to the light-dark cycle and releases melatonin primarily during darkness, regulating sleep-wake cycles. 7. Gonads, ovaries, and testes. Gonadotropin regulation. GnRH from the hypothalamus stimulates LH and FSH release from the anterior pituitary, which regulates sex hormone secretion from the gonads. 8. Hypothalamus. Feedback regulation. Feedback from target gland hormones inhibits hypothalamic releasing hormone secretion. 9. Other regulatory mechanisms. Feedback loops. Hormones often exert feedback regulation, inhibiting their own secretion. For instance, elevated cortisol levels inhibit CRH and ACTH release. Circadian rhythms. Hormone secretion often follows daily patterns influenced by the body's internal clock. The regulation of hormone secretion involves a complex interplay of feedback loops, neural inputs, environmental factors, and interactions between multiple endocrine glands. The goal is to maintain homeostasis and ensure the body's various systems function optimally. Pathophysiology of endocrine glands in detail. Very long answer. ChatGPT. Certainly, let's explore the pathophysiology of some key endocrine glands in detail. 1. Thyroid gland. Hypothyroidism. Insufficient thyroid hormone production leads to symptoms such as fatigue, weight gain, and cold sensitivity. Causes include Hashimoto's thyroiditis, iodine deficiency, and certain medications. Hyperthyroidism. Excessive thyroid hormone production results in symptoms like weight loss, rapid heartbeat, and heat intolerance. Hypothyroidism. Insufficient thyroid hormone production. Hypothyroidism. Insufficient thyroid hormone production leads to symptoms such as fatigue, weight gain, and cold sensitivity. Causes include Hashimoto's thyroiditis, iodine deficiency, and certain medications. Hyperthyroidism. Excessive thyroid hormone production results in symptoms like weight loss, rapid heartbeat, and heat intolerance. Causes include Graves' disease and toxic adenoma. 2. Adrenal glands. Adrenal insufficiency. Hypothyroidism. Insufficient thyroid hormone production leads to symptoms such as fatigue, weight gain, and cold sensitivity. Causes include Hashimoto's thyroiditis, iodine deficiency, and certain medications. Hyperthyroidism. Excessive thyroid hormone production results in symptoms like weight loss, rapid heartbeat, and heat intolerance. Causes include Graves' disease and toxic adenoma. 2. Adrenal glands. Adrenal insufficiency. Addison's disease. Insufficient adrenal hormones, cortisol and aldosterone, led to fatigue, weight loss, low blood pressure, and electrolyte imbalances. Autoimmune destruction, infections, or medication can cause this. Cushing's syndrome. Excess cortisol production causes weight gain, muscle weakness, and hypertension. This can result from adrenal tumors or excessive use of glucocorticoid medications. 3. Pancreas. Type 1 diabetes. Autoimmune destruction of pancreatic beta cells leads to insulin deficiency. High blood sugar levels cause polyuria, polydipsia, and weight loss. Type 2 diabetes. Insulin resistance and inadequate insulin secretion result in high blood sugar levels. Obesity, genetics, and lifestyle contribute. 4. Parathyroid glands. Hyperparathyroidism. Overactive parathyroid glands lead to high blood calcium levels, causing fatigue, kidney stones, and osteoporosis. Hypoparathyroidism. Low parathyroid hormone levels result in low blood calcium, causing muscle cramps, tingling, and seizures. 5. Pituitary gland. Pituitary tumors. These can result in hormone overproduction, e.g., acromegaly due to excess GH, or underproduction, e.g., hypopituitarism due to insufficient ACTH or LH-FSH. 6. Gonads, ovaries, and testes. Polycystic ovary syndrome (PCOS). Elevated androgen levels cause irregular periods, hirsutism, and infertility in females. Hypogonadism. Reduced sex hormone production leads to delayed puberty, infertility, and low libido. 7. Pineal gland. Melatonin imbalance. Disrupted melatonin production can result from altered sleep patterns, e.g., shift work, and affect sleep-wake cycles. 8. Hormone resistance syndromes. Insulin resistance syndrome. Melatonin imbalance. Disrupted melatonin production can result from altered sleep patterns, e.g., shift work, and affect sleep-wake cycles. 8. Hormone resistance syndromes. Insulin resistance syndrome. Cells become resistant to insulin, leading to type 2 diabetes, obesity, and cardiovascular risk factors. Androgen insensitivity syndrome. Genetic mutation results in inability of cells to respond to androgens, leading to varying degrees of feminization in individuals with XY chromosomes. Pathophysiological conditions can stem from genetic mutations, autoimmune responses, tumors, infections, and environmental factors. Diagnosis often involves blood tests, imaging, and hormone level measurements. Treatment varies based on the specific condition and may involve medications, hormone replacement, surgery, or lifestyle changes. Hypothalamus. Anatomy, structure, physiological functions, control of secretion of hormones, and pathophysiology. Each detail. Very, very long answer. ChatGPT. Hypothalamus. Anatomy, structure, physiological functions, control of hormone secretion, and pathophysiology. Anatomy and structure. The hypothalamus is a small region located at the base of the brain, above the pituitary gland. It consists of several nuclei, each with specific functions related to regulating homeostasis, behavior, and endocrine function. Key nuclei include the paraventricular nucleus (PVN), supraoptic nucleus, sun-arcuate nucleus, and ventromedial nucleus. Physiological functions. Homeostasis regulation. The hypothalamus plays a central role in maintaining various physiological parameters such as body temperature, hunger, thirst, and fluid balance. Endocrine control. The hypothalamus controls the release of hormones from the pituitary gland, which in turn regulates other endocrine glands. Circadian rhythms. The suprachiasmatic nucleus (SCN) of the hypothalamus helps regulate the body's internal clock, influencing sleep-wake cycles. Autonomic nervous system. The hypothalamus controls the autonomic nervous system, affecting processes like heart rate, blood pressure, and digestion. Emotional responses. The hypothalamus influences emotions and behaviors through connections with the limbic system. Control of hormone secretion. The hypothalamus produces releasing and inhibiting hormones that control the anterior pituitary gland's hormone secretion. Hypothalamic releasing hormones, e.g., TRH, CRH, GnRH, stimulate the pituitary to release hormones like thyroid-stimulating hormone, TSH, adrenocorticotropic hormone, ACTH, and gonadotropins, LH and FSH. Feedback loops involving hormones from peripheral glands regulate hypothalamic hormone secretion. Pathophysiology. Hypothalamic dysfunction. Tumors, infections, or injuries can disrupt hypothalamic function, leading to hormone imbalances, obesity, and temperature dysregulation. Hypothalamic pituitary axis disorders. Dysregulation of hormone secretion from the hypothalamus and pituitary can lead to conditions like Cushing's disease or hypopituitarism. Endocrine disorders. Hypothalamic dysfunction can impact other endocrine glands, causing conditions like hypothyroidism or growth hormone deficiency. Sleep disorders. Disruption of the hypothalamic SCN can lead to sleep-wake cycle disorders like insomnia or jet lag. Eating disorders. Hypothalamic involvement in appetite regulation contributes to conditions like anorexia and obesity. In summary, the hypothalamus is a critical brain region that integrates various physiological processes, controls hormone secretion, and influences behavior. Dysregulation of its functions can lead to a range of complex endocrine, neurological, and behavioral disorders. Hypothesis. Anatomy, structure, physiological functions, control of secretion of hormones, and pathophysiology. Each detail. Very, very long answer. ChatGPT. Hypothesis. Pituitary gland anatomy, structure, physiological functions, control of hormone secretion, and pathophysiology. Anatomy and structure. The pituitary gland, or hypophysis, is a small pea-sized gland located at the base of the brain, beneath the hypothalamus, within a bony structure called the sella turcica. It is divided into two major parts: the anterior pituitary adenohypophysis and the posterior pituitary neurohypophysis. The anterior pituitary is glandular and synthesizes and releases hormones, while the posterior pituitary stores and releases hormones produced by the hypothalamus. Physiological functions. Anterior pituitary functions. The anterior pituitary secretes hormones that regulate various endocrine glands and physiological processes, including growth, metabolism, reproduction, and stress response. Posterior pituitary functions. The posterior pituitary releases hormones produced by the hypothalamus, including oxytocin, stimulates uterine contractions and milk ejection, and vasopressin controls water balance and blood pressure. Control of hormone secretion. Hypothalamus pituitary axis. The hypothalamus controls hormone release from the anterior pituitary through releasing and inhibiting hormones. Anterior pituitary hormones. Hormones produced by the anterior pituitary include growth hormone GH, adrenocorticotropic hormone ACTH, thyroid stimulating hormone TSH, prolactin PRL, follicle stimulating hormone FSH, and luteinizing hormone LH. Feedback loops. Peripheral glands, such as the thyroid or adrenal glands, produce hormones that feedback to regulate the release of anterior pituitary hormones. Pathophysiology. Hypopituitarism. Dysfunction of the pituitary gland can lead to inadequate hormone production, resulting in conditions like growth hormone deficiency, thyroid dysfunction, and adrenal insufficiency. Pituitary tumors. Abnormal growths in the pituitary can lead to hormone overproduction or underproduction, affecting various target tissues. Hyperpituitarism. Excessive hormone secretion from the pituitary can cause conditions like acromegaly, GHXS, or Cushing's disease, ACTHXS. Diabetes insipidus. Dysfunction of the posterior pituitary can result in inadequate secretion of vasopressin, leading to excessive urination and thirst. Sheehan's syndrome. Pregnancy-related blood loss can lead to pituitary infarction, causing hypopituitarism. Calcium regulation. The primary function of the parathyroid glands is to regulate calcium levels in the blood. Parathyroid hormone PTH production. The parathyroid glands produce parathyroid hormone PTH, which plays a crucial role in calcium homeostasis. Bone remodeling. PTH stimulates bone resorption, releasing calcium and phosphate into the bloodstream. Kidney function. PTH acts on the kidneys to increase calcium reabsorption while promoting phosphate excretion. Control of hormone secretion. Calcium sensing. The parathyroid glands sense blood calcium levels and respond by releasing PTH. Negative feedback loop. When blood calcium levels decrease, the parathyroid glands release PTH to increase calcium levels. Elevated calcium levels suppress PTH release. Pathophysiology. Hyperparathyroidism. Overactivity of the parathyroid glands leads to excessive PTH production. Primary hyperparathyroidism can result from benign tumors, adenoma, or hyperplasia of the parathyroid glands. It causes hypercalcemia, leading to kidney stones, bone loss, and other complications. Hypoparathyroidism. Reduced PTH production leads to hypocalcemia and impaired calcium regulation. It can result from surgical removal of the parathyroid glands or autoimmune disorders. Parathyroid adenoma. Benign tumors of the parathyroid glands can cause hyperparathyroidism. Surgical removal is often necessary. Parathyroid carcinoma. Rare malignant tumors of the parathyroid glands can cause hyperparathyroidism and require aggressive treatment. Renal disease and secondary hyperparathyroidism. Chronic kidney disease can lead to secondary hyperparathyroidism due to impaired calcium and phosphate regulation. In conclusion, the parathyroid glands play a critical role in maintaining calcium homeostasis, bone health, and kidney function. Disorders of the parathyroid glands can lead to imbalances in calcium levels and have significant effects on overall health. Adrenal, anatomy, structure, physiological functions, control of secretion of hormones, and pathophysiology. Each detail very long answer. ChatGPT. Adrenal glands, anatomy, structure, physiological functions, control of hormone secretion, and pathophysiology. Anatomy and structure. The adrenal glands are paired endocrine glands located on top of each kidney. Each adrenal gland is divided into two main parts, the outer adrenal cortex and the inner adrenal medulla. Physiological functions. Adrenal cortex functions. Mineralocorticoids. The outermost layer produces mineralocorticoids, mainly aldosterone, which regulates electrolyte and water balance. Glucocorticoids. The middle layer produces glucocorticoids, mainly cortisol, which regulates metabolism, immune response, and stress response. Androgens. The innermost layer produces androgens, including DHEA and androstenedione, which have weak masculinizing effects. Adrenal medulla function. The adrenal medulla produces catecholamines, such as adrenaline, epinephrine, and noradrenaline, norepinephrine, which are involved in the fight or flight stress response. Control of hormone secretion. Adrenal cortex regulation. The hypothalamus releases corticotropin-releasing hormone (CRH), which stimulates the anterior pituitary to release adrenocorticotropic hormone (ACTH). ACTH then stimulates the adrenal cortex to release cortisol. Adrenal medulla regulation. The sympathetic nervous system triggers the release of catecholamines, adrenaline, and noradrenaline from the adrenal medulla during stress. Pathophysiology. Hyperaldosteronism. Overproduction of aldosterone can lead to hypertension, fluid retention, and electrolyte imbalances. Cushing's syndrome. Excessive cortisol production, whether due to adrenal tumors or long-term glucocorticoid therapy, leads to weight gain, metabolic disturbances, and immunosuppression. Addison's disease. Insufficient cortisol and often aldosterone production due to adrenal gland damage results in fatigue, weight loss, and electrolyte imbalances. Pheochromocytoma. Adrenal medulla tumors can cause excessive catecholamine release, leading to hypertension, rapid heart rate, and anxiety. Congenital adrenal hyperplasia. Genetic enzyme deficiencies can disrupt adrenal hormone synthesis, leading to abnormal sex development and metabolic imbalances. The adrenal glands play essential roles in maintaining homeostasis, responding to stress, and regulating various physiological processes. Dysregulation of adrenal hormone production can have far-reaching effects on overall health and well-being. Pancreas, anatomy, structure, physiological functions, control of secretion of hormones, and pathophysiology. Each detail very long answer. ChatGPT. Pancreas, anatomy, structure. Pancreas, anatomy, structure, physiological functions, control of hormone secretion, and pathophysiology. Anatomy and structure. The pancreas is a mixed gland located behind the stomach, composed of both endocrine and exocrine tissues. The exocrine part consists of acinar cells that produce digestive enzymes, while the endocrine part consists of clusters of cells called pancreatic islets, islets of Langerhans. Physiological functions. Exocrine function. Acinar cells secrete digestive enzymes amylase, lipase, proteases into the pancreatic duct, which joins the common bile duct and empties into the duodenum to aid in digestion. Endocrine function. Pancreatic islets contain different types of cells. Alpha cells secrete glucagon, which raises blood glucose levels by stimulating the liver to release glucose. Beta cells secrete insulin, which lowers blood glucose levels by promoting glucose uptake into cells and inhibiting glucose release from the liver. Delta cells secrete somatostatin, which inhibits the release of both insulin and glucagon. PP cells secrete pancreatic polypeptide, which regulates pancreatic and gastrointestinal functions. Control of hormone secretion. Insulin secretion. Elevated blood. Alpha cells secrete glucagon, which raises blood glucose levels by stimulating the liver to release glucose. Beta cells secrete insulin, which lowers blood glucose levels by promoting glucose uptake into cells and inhibiting glucose release from the liver. Delta cells secrete somatostatin, which inhibits the release of both insulin and glucagon. PP cells secrete pancreatic polypeptide, which regulates pancreatic and gastrointestinal functions. Control of hormone secretion. Insulin secretion. Elevated blood glucose levels stimulate beta cells to release insulin. Glucagon secretion. Decreased blood glucose levels or increased amino acid levels stimulate alpha cells to release glucagon. Somatostatin secretion. Released in response to both insulin and glucagon to regulate their secretion. Pathophysiology. Type 1 diabetes. Autoimmune destruction of beta cells leads to insulin deficiency, causing hyperglycemia, polyuria, weight loss, and ketoacidosis. Type 2 diabetes. Insulin resistance and impaired beta cell function result in elevated blood glucose levels, leading to similar symptoms as type 1 diabetes. Pancreatitis. Inflammation of the pancreas can cause severe abdominal pain, digestive enzyme leakage, and potential damage to the islets, affecting insulin and glucagon secretion. Pancreatic cancer. Tumors in the pancreas can disrupt both exocrine and endocrine functions, leading to digestive problems and hormonal imbalances. The pancreas plays a crucial role in regulating glucose levels in digestive processes. Dysregulation of its functions can lead to serious metabolic and digestive disorders, making its proper function essential for overall health. Hormones and male reproductive physiology in detail. Very long answer. ChatGPT. Hormones and male reproductive physiology. Hormones. Gonadotropin-releasing hormone, GnRH. Secreted by the hypothalamus. Stimulates the anterior pituitary to release luteinizing hormone, LH, and follicle-stimulating hormone, FSH. Luteinizing hormone, LH. Released by the anterior pituitary. Stimulates Leydig cells in the testes to produce testosterone. Follicle-stimulating hormone, FSH. Released by the anterior pituitary. Stimulates Sertoli cells in the testes to support spermatogenesis. Testosterone. Produced by Leydig cells in the testes. Promotes the development of secondary sexual characteristics, sperm production, and maintains male reproductive organs. Inhibin. Released by Sertoli cells. Inhibits FSH secretion through negative feedback on the pituitary. Physiological functions. Spermatogenesis. FSH stimulates Sertoli cells to support the maturation of sperm cells. Testosterone is required for the progression of spermatogenesis. Secondary sexual characteristics. Testosterone induces the development of facial hair, deepening of the voice, muscle growth, and enlargement of the penis and testes. Libido and sexual function. Testosterone plays a role in maintaining sexual desire and erectile function. Prostate health. Androgens like testosterone contribute to the growth and maintenance of the prostate gland. Negative feedback mechanism. High levels of testosterone inhibit GnRH and LH secretion via negative feedback, regulating the hormonal balance. Regulation. Hypothalamic pituitary gonadal HPG axis. GnRH from the hypothalamus stimulates the release of LH and FSH from the anterior pituitary. LH stimulates Leydig cells to produce testosterone. FSH stimulates Sertoli cells to support spermatogenesis. Negative feedback loop. Elevated testosterone levels inhibit GnRH and LH secretion through negative feedback. Pathophysiology. Hypogonadism. Insufficient testosterone production leads to reduced sperm count, infertility, decreased libido, and muscle loss. Andropause, age-related testosterone decline. Gradual decrease in testosterone levels with aging can lead to reduced libido, fatigue, and mood changes. Testicular disorders. Conditions like testicular cancer, varicocele, and mumps orchitis can affect testosterone production and spermatogenesis. Hormone imbalances. Disorders in the HPG axis can lead to hormonal imbalances, affecting reproductive health. Understanding the hormonal regulation of male reproductive physiology is crucial for maintaining reproductive health, fertility, and overall well-being. Hormones like testosterone play a vital role in the development of male characteristics, sexual function, and the production of healthy sperm. Synthesis of androgens in detail. Very long answer. ChatGPT. Synthesis of androgens. Androgens are a class of steroid hormones that include testosterone and dihydrotestosterone (DHT). These hormones play a crucial role in the development and maintenance of male reproductive organs, secondary sexual characteristics, and overall male physiology. The synthesis of androgens occurs primarily in the testes, under the control of the hypothalamic pituitary gonadal HPG axis. Step-by-step synthesis of androgens. Stimulation of the hypothalamus. Gonadotropin-releasing hormone (GnRH) is secreted by the hypothalamus in response to hormonal signals and environmental cues. Pituitary stimulation. Conversion of gonadotropin-releasing hormone (GnRH) is secreted by the hypothalamus in response to hormonal signals and environmental cues. Pituitary stimulation. GnRH reaches the anterior pituitary and stimulates the release of luteinizing hormone (LH) and follicle-stimulating hormone (FSH). Leydig cell activation. LH travels to the testes and binds to Leydig cells, stimulating their activation. Cholesterol uptake. Leydig cells possess enzymes that allow them to take up cholesterol from circulating lipoproteins. Conversion of cholesterol to pregnenolone. Cholesterol is converted into pregnenolone through a series of enzymatic reactions, including desmolase. Conversion of pregnenolone to progesterone. Pregnenolone is converted into progesterone through various enzymatic reactions. Conversion of progesterone to androgens. Progesterone is further converted into androgens through a series of enzymatic reactions. The key enzyme 17-alpha-hydroxylase converts progesterone to 17-alpha-hydroxyprogesterone. Conversion of androgens to testosterone. Androstenedione, one of the androgens produced, is further converted into testosterone by the enzyme 17-beta-hydroxysteroid dehydrogenase. Conversion of testosterone to dihydrotestosterone (DHT). Testosterone can be converted to DHT in target tissues by the enzyme 5-alpha reductase. DHT is a more potent androgen, responsible for the development of male secondary sexual characteristics. Control of androgen synthesis. The synthesis of androgens is regulated by a negative feedback mechanism to maintain hormonal balance. GnRH regulation. Elevated testosterone levels inhibit the secretion of GnRH from the hypothalamus, leading to decreased LH and FSH secretion. LH and FSH regulation. Elevated testosterone levels inhibit LH secretion, reducing Leydig cell activation and androgen synthesis. FSH stimulates Sertoli cells to support spermatogenesis and indirectly influences androgen production. Pathophysiology. Disruptions in the synthesis of androgens can lead to various health issues. Hypogonadism. Insufficient androgen production can lead to hypogonadism, causing reduced libido, fatigue, and infertility. Androgen excess. Conditions like polycystic ovary syndrome (PCOS) can lead to excessive androgen production in females, causing irregular periods and other symptoms. Understanding the detailed synthesis of androgens is crucial for comprehending the physiological processes that underlie male reproductive health, secondary sexual characteristics, and overall male well-being. Chemistry of androgens in detail. Very long answer. ChatGPT. Chemistry of androgens. Androgens are a class of steroid hormones that play a pivotal role in the development and maintenance of male reproductive organs, secondary sexual characteristics, and various physiological processes. The chemistry of androgens revolves around their structure, synthesis, and interactions within the body. Steroid hormone structure. Androgens, like other steroid hormones, share a common structure characterized by a tetracyclic carbon skeleton composed of 3 cyclohexane rings, labeled A, B, and C, and 1 cyclopentane ring, labeled D. The rings are fused together and are designated as A, B, B, C, and C, D rings. The structure also includes side chains and functional groups that determine the hormone's biological activity. Cholesterol precursor. Androgens are synthesized from cholesterol, which is obtained from circulating lipoproteins. Cholesterol serves as the starting material for the synthesis of androgens in the Leydig cells of the testes. Key enzymes. Several key enzymes are involved in the synthesis of androgens, including desmolase, converts cholesterol to pregnenolone, a precursor of all steroid hormones. 17-alpha-hydroxylase, converts pregnenolone to 17-alpha-hydroxyprogesterone. 17-beta-hydroxysteroid dehydrogenase, converts androstenedione to testosterone. 5-alpha reductase, converts testosterone to the more potent dihydrotestosterone (DHT). Functional groups and side chains. The functional groups and side chains of androgens contribute to their specific actions and interactions. Testosterone and DHT, for instance, both have hydroxyl groups O at the 17 beta position. DHT has an additional double bond between the fourth and fifth carbon atoms, making it more potent than testosterone in terms of androgenic effects. Androgen receptors. Androgens exert their effects by binding to androgen receptors, ARs, which are located in various tissues throughout the body. The binding of androgens to ARs triggers a series of intracellular events that regulate gene expression and mediate the physiological responses associated with androgens. Biological effects. Androgens have a broad range of biological effects, including promotion of male secondary sexual characteristics, e.g., facial hair, deep voice, regulation of sexual function, libido, and sperm production, maintenance of muscle mass and bone density, influence on mood, energy levels, and overall well-being, pathophysiology. Disruptions in androgen chemistry can lead to various health conditions. For example, deficiencies in androgen production can result in hypogonadism, characterized by reduced sexual development and other associated symptoms. Androgen excess, on the other hand, can lead to conditions like hirsutism, excessive hair growth, and masculinization in females. Understanding the chemistry of androgens is essential for comprehending their role in male physiology, development, and health. Understanding the chemistry of androgens is essential for comprehending their role in male physiology, development, and health. It also sheds light on the potential therapeutic interventions for conditions related to androgen imbalance. Metabolism of androgens in detail. Very long answer. ChatGPT. Metabolism of androgens. The metabolism of androgens, such as testosterone and dihydrotestosterone (DHT), is a complex process involving various enzymes, tissues, and pathways. Understanding the metabolism of androgens is crucial as it impacts their bioavailability, activity, and overall physiological effects. Testosterone metabolism. Synthesis. Testosterone is synthesized primarily in the Leydig cells of the testes. It is then released into the bloodstream, bound to sex hormone-binding globulin (SHBG) and albumin. Tissue uptake. Testosterone can enter target tissues, where it binds to androgen receptors (ARs) and exerts its effects. It can also be converted to DHT within target tissues, such as the prostate and skin, by the enzyme 5-alpha reductase. Metabolism in liver. Testosterone that is not bound to SHBG or albumin is taken up by the liver, where it is then released into the bloodstream, bound to sex hormone-binding globulin (SHBG) and albumin. Tissue uptake. Testosterone can enter target tissues, where it binds to androgen receptors (ARs) and exerts its effects. It can also be converted to DHT within target tissues, such as the prostate and skin, by the enzyme 5-alpha reductase. Metabolism in liver. Testosterone that is not bound to SHBG or albumin is taken up by the liver, where it undergoes metabolism. In the liver, testosterone is transformed into inactive metabolites such as androsterone and acetylanolone, which are conjugated with glucuronic acid or sulfate for excretion. Dihydrotestosterone (DHT) metabolism. Conversion from testosterone. DHT is synthesized from testosterone in target tissues by the enzyme 5-alpha reductase. This enzyme catalyzes the conversion of the 4,5 double bond of testosterone to form DHT, which is more potent than testosterone in terms of androgenic effects. Tissue effects. DHT binds to ARs with higher affinity than testosterone and remains active for a longer period. This makes DHT a critical androgen in tissues such as the prostate, where it contributes to the development and maintenance of male secondary sexual characteristics. Metabolic regulation. The metabolism of androgens is regulated by feedback mechanisms involving the hypothalamus, pituitary gland, and testes. The hypothalamus releases gonadotropin-releasing hormone (GnRH), which stimulates the anterior pituitary to release luteinizing hormone (LH) and follicle-stimulating hormone (FSH). LH, in particular, stimulates Leydig cells in the testes to produce testosterone. Clinical implications. Alterations in androgen metabolism can lead to various clinical conditions. Hirsutism, excessive androgen production, often due to polycystic ovary syndrome (PCOS) or adrenal disorders, can cause increased hair growth in women. Virilization. In women, excessive androgens can lead to masculinizing features, such as deepening of the voice and enlargement of the clitoris. Androgen insensitivity syndrome. Genetic mutations in the AR gene can result in a lack of response to androgens, leading to incomplete or absent masculinization in XY individuals. Conclusion. The metabolism of androgen insensitivity syndrome. Genetic mutations in the AR gene can result in a lack of response to androgens, leading to incomplete or absent masculinization in XY individuals. Conclusion. The metabolism of androgens is a dynamic process influenced by multiple factors. It involves the synthesis, conversion, and degradation of hormones to ensure proper physiological effects while preventing excessive androgen exposure. Understanding this intricate process is essential for diagnosing and managing conditions related to androgen metabolism. Synthesis, chemistry, and metabolism of androgens in detail. ChatGPT. Synthesis, chemistry, and metabolism of androgens. Synthesis. Androgens are a class of steroid hormones primarily produced by the testes in males and in smaller amounts by the adrenal glands in both males and females. The main androgen is testosterone, which plays a crucial role in the development and maintenance of male reproductive organs and secondary sexual characteristics. Leydig cells. In the testes, Leydig cells synthesize testosterone through a series of enzymatic reactions known as steroidogenesis. The process is regulated by luteinizing hormone (LH) secreted by the anterior pituitary gland in response to gonadotropin-releasing hormone (GnRH) from the Leydig cells. In the testes, Leydig cells synthesize testosterone through a series of enzymatic reactions known as steroidogenesis. The process is regulated by luteinizing hormone (LH) secreted by the anterior pituitary gland in response to gonadotropin-releasing hormone. The process is regulated by luteinizing hormone (LH) secreted by the anterior pituitary gland in response to gonadotropin-releasing hormone (GnRH) from the hypothalamus. Chemistry. Testosterone is a C19 steroid hormone with a specific chemical structure. Its chemical formula is C19H28O2, and it belongs to the androstenedione class of steroids. Testosterone's structure consists of 4 interconnected rings, 3 cyclohexane rings, and 1 cyclopentane ring, and a hydroxyl group at the 17th carbon. Metabolism. Testosterone metabolism involves both peripheral tissues and the liver. The main pathways include conversion to dihydrotestosterone (DHT) in target tissues such as the prostate, skin, and hair follicles. Testosterone is converted to DHT by the enzyme 5-alpha reductase. DHT is more potent than testosterone and is responsible for the development of male secondary sexual characteristics. Aromatization. Some testosterone is converted to estradiol and estrogen through the process of aromatization. This occurs mainly in adipose tissue, liver, and the brain, where the enzyme aromatase converts testosterone to estradiol. This estrogenic conversion has important implications for both males and females. Metabolism in the liver. In the liver, testosterone undergoes metabolism through phase 1 and phase 2 reactions. It is conjugated with glucuronic acid or sulfate, making it water-soluble and facilitating its excretion in the urine. Clinical implications. Hypogonadism. Insufficient androgen production can lead to hypogonadism, resulting in decreased libido, muscle mass, and bone density. Androgen replacement therapy. Testosterone replacement therapy is used to treat hypogonadism. It can improve muscle strength, bone density, and quality of life. Androgen-related disorders. Conditions such as polycystic ovary syndrome (PCOS) and androgen insensitivity syndrome (ICE) are characterized by altered androgen levels and responsiveness. Prostate health. DHT is implicated in the development of benign prostatic hyperplasia (BPH) and prostate cancer. Medications that inhibit 5-alpha reductase are used to manage these conditions. Conclusion. The synthesis, chemistry, and metabolism of androgens, particularly testosterone, are intricate processes that play a central role in male development, reproduction, and overall health. Understanding these processes is vital for diagnosing and managing various hormonal imbalances and related disorders. Endocrine control of testicular function in detail. Very long answer. ChatGPT. Endocrine control of testicular function. The testes are vital male reproductive organs responsible for the production of sperm and androgens, primarily testosterone. The endocrine control of testicular function involves a complex interplay of hormones from various endocrine glands. This process is essential for the development of secondary sexual characteristics, maintenance of reproductive organs, and overall male health. Hypothalamus pituitary testes axis. Hypothalamus. The process begins in the hypothalamus, which secretes gonadotropin-releasing hormone GnRH. GnRH stimulates the anterior pituitary gland to release the gonadotropins, luteinizing hormone LH, and follicle-stimulating hormone FSH. Anterior pituitary gland, LH and FSH act on the Leydig cells and Sertoli cells of the testes, respectively. Anterior pituitary gland, LH and FSH act on the Leydig cells and Sertoli cells of the testes, respectively. Leydig cells. Luteinizing hormone LH binds to receptors on Leydig cells, stimulating the production of testosterone through a series of enzymatic reactions called steroidogenesis. Testosterone is the primary androgen responsible for male sexual development and maintenance. Sertoli cells. Follicle-stimulating hormone FSH binds to receptors on Sertoli cells, supporting spermatogenesis, which is the process of sperm production. Sertoli cells provide nourishment and structural support for developing sperm cells. Testosterone feedback loop. Negative feedback. As testosterone levels rise, they exert negative feedback on the hypothalamus and pituitary, inhibiting the release of GnRH and gonadotropins. This maintains hormonal homeostasis. Additional hormones involved. Inhibin, produced by Sertoli cells. Inhibin inhibits the secretion of FSH from the anterior pituitary. It serves as a local feedback mechanism to regulate spermatogenesis. Prolactin, although not directly involved in the hypothalamus pituitary testes axis, elevated prolactin levels can suppress gonadotropin secretion and impair testicular function. Clinical implications. Hypogonadism. Inadequate testosterone production can lead to hypogonadism, resulting in reduced libido, muscle mass, and bone density. Testosterone replacement therapy can be used to address hypogonadism. Infertility. Hormonal imbalances can negatively impact sperm production and quality. Understanding the endocrine control of testicular function is crucial for diagnosing and treating male infertility. Hormone therapy. Endocrine control is often manipulated in assisted reproductive technologies to enhance sperm production and quality, thereby improving the chances of successful fertilization. Androgen-related disorders. Disturbances in the endocrine control of testicular function can lead to conditions such as Klinefelter syndrome, in which individuals have an extra X chromosome, affecting testicular development and testosterone production. Conclusion. The endocrine control of testicular function is a finely tuned process involving interactions between the hypothalamus, pituitary gland, and testes. Hormones such as GnRH, LH, FSH, testosterone, and inhibin play critical roles in the regulation of spermatogenesis, testosterone production, and overall male reproductive health. Understanding this complex axis is essential for diagnosing and managing various male reproductive disorders and optimizing reproductive outcomes. Physiological roles of androgens in detail. Very long answer. ChatGPT. Physiological roles of androgens. Androgens are a class of male sex hormones that play diverse and essential roles in the development and maintenance of male reproductive and secondary sexual characteristics. The primary androgen is testosterone, produced primarily in the testes and to a lesser extent in the adrenal glands. These hormones exert their effects through binding to androgen receptors in target tissues. The physiological roles of androgens encompass various aspects of growth, development, metabolism, and sexual function. 1. Development of secondary sexual characteristics. Androgens are responsible for the development of male secondary sexual characteristics during puberty. These include the growth of facial and body hair, deepening of the voice, and increased muscle mass. 2. Regulation of muscle mass and strength. Androgens, particularly testosterone, promote the growth of muscle tissue by stimulating protein synthesis. This is why males generally have greater muscle mass and strength compared to females. 3. Bone health and density. Androgens play a crucial role in maintaining bone health by promoting bone mineralization and density. They also contribute to the closure of epiphyseal plates, leading to cessation of longitudinal bone growth. 4. Hematopoiesis. They also contribute to the closure of epiphyseal plates, leading to cessation of longitudinal bone growth. 4. Hematopoiesis. Androgens influence the production. Androgens influence the production of red blood cells, erythropoiesis, by stimulating erythropoietin production in the kidneys. This contributes to the higher red blood cell count in males compared to females. 5. Libido and sexual function. Androgens, particularly testosterone, are critical for the maintenance of libido, sexual desire, and overall sexual function. They influence sexual arousal, erectile function, and the production of sperm. 6. Regulation of fat distribution. Androgens influence fat distribution in the body, helping to maintain a more centralized pattern of fat accumulation in males, often referred to as android or apple body shape. 7. Cognitive function. Androgens are thought to play a role in cognitive function, mood regulation, and behavioral pattern. Androgens are thought to play a role in cognitive function, mood regulation, and behavioral patterns. Some studies suggest that testosterone may impact spatial and mathematical abilities. 8. Metabolism. Androgens can influence metabolism by affecting insulin sensitivity, glucose metabolism, and lipid metabolism. They may contribute to a lower tendency for fat accumulation and obesity in males. 9. Skin health. Androgens contribute to the regulation of sebum production in the skin's sebaceous glands. This can impact skin health and the development of acne during puberty. 10. Cardiovascular health. Androgens may have a protective effect on cardiovascular health, including improving vascular function and reducing the risk of cardiovascular diseases. 11. Mood and well-being. Some studies suggest a link between testosterone levels and mood. Low levels of testosterone may be associated with mood disorders like depression. 12. Brain development. Androgens play a role in brain development, influencing the sexual differentiation of the brain during fetal development. Clinical implications. Androgen-related disorders, such as hypogonadism, low testosterone levels, and androgen excess syndromes, can lead to various health issues. Hormone replacement therapy can be used to address hormonal imbalances and restore normal physiological functions. Conclusion. Androgens, particularly testosterone, play diverse and critical roles in male growth, development, metabolism, sexual function, and overall well-being. Their effects extend beyond sexual characteristics and encompass various physiological systems, contributing to the maintenance of male health throughout life. Physiological roles of estrogens in detail. Very long answer. ChatGPT. Physiological roles of estrogens. Estrogens are a group of steroid hormones that play vital roles in the development and maintenance of female reproductive and secondary sexual characteristics. The primary endogenous estrogens are estradiol, estrone, and estriol, with estradiol being the most potent. Estrogens are a group of steroid hormones that play vital roles in the development and maintenance of female reproductive and secondary sexual characteristics. The primary endogenous estrogens are estradiol, estrone, and estriol, with estradiol being the most potent. These hormones are produced primarily in the ovaries, as well as in smaller amounts in other tissues like the adrenal glands, fat cells, and placenta during pregnancy. These hormones are produced primarily in the ovaries, as well as in smaller amounts in other tissues like the adrenal glands, fat cells, and placenta during pregnancy. Estrogens exert their effects through binding to estrogen receptors in target tissues. The physiological roles of estrogens are diverse and encompass various aspects of growth, development, reproduction, and overall health. 1. Development of secondary sexual characteristics. Estrogens are responsible for the development of female secondary sexual characteristics during puberty. These include breast development, widening of the hips, and changes in body fat distribution. 2. Regulation of menstrual cycle. Estrogens, particularly estradiol, are essential for the regulation of the menstrual cycle. They stimulate the growth and development of the uterine lining endometrium in preparation for potential pregnancy. 3. Reproductive function. Estrogens play a crucial role in the development and maintenance of the female reproductive system. They promote the growth and maturation of ovarian follicles, which contain developing eggs. 4. Bone health and density. Estrogens are vital for maintaining bone health by promoting bone mineralization and inhibiting bone resorption. Estrogen deficiency, as seen in menopause, can lead to decreased bone density and increased risk of osteoporosis. 5. Cardiovascular health. Estrogens may have a protective effect on cardiovascular health. They can help maintain healthy blood vessels, regulate cholesterol levels, and reduce the risk of cardiovascular diseases. 6. Skin health. Estrogens influence skin health by promoting collagen production and maintaining skin hydration. They contribute to the overall texture and appearance of the skin. 7. Mood regulation. Estrogens may influence mood and emotional well-being. Some studies suggest that fluctuations in estrogen levels throughout the menstrual cycle can impact mood. 8. Cognitive function. Estrogens are thought to play a role in cognitive function and memory. They may have neuroprotective effects on the brain. 9. Libido and sexual function. Estrogens contribute to sexual desire and overall sexual function in females. They influence vaginal lubrication and the health of vaginal tissues. 10. Metabolism. Estrogens can influence metabolism by affecting insulin sensitivity and glucose metabolism. They may contribute to the lower tendency for central fat accumulation in females. 11. Pregnancy and development.